I'm assuming they can. Okay, so today we're very excited to welcome Hamid, Karimi, Rouzbahani. So Hamid has a background as an engineer and does work at the intersection of cognitive science, biomedical engineering, and machine learning. And I got to know Hamid when we were both at the Cognition Brain Sciences unit in Cambridge, where he was a Newton Fellow. Now in Australia, Hamid splits his time between the Mater Hospital as part of the Epilepsy group and the Queensland Brain Institute, where he's a DECRA Fellow. And so we're very excited today to hear about some recent work that I think is very relevant to many of the people in the SNAC group, using PySpy to characterize the directionality of neural activity at seizure onset. Okay, I'll turn it over to Hamid.
Thank you, Chris, for the introduction, and thank you for the opportunity to present my work. So, as Chris mentioned, I'm working as a biomedical engineer at Mater Hospital's Epilepsy group as well, which is where I worked on these projects which I'm presenting today. So we are focused on epilepsy in Mater, and the outline of my talk is I will give an overview of epilepsy and the localization problem in epilepsy. Then I will present an overview of two of my studies. The first one is on the localization of the epileptogenic zone and the generalizability of epileptic patterns across time and patients. And the second one is on the flow of neural activity as a biomarker of epileptogenic zone localization. So, introduction, I think the take-home message from my talk will be this one. We tend to look at a phenomenon from different angles, and we get different samples of the phenomenon. For example, looking at an elephant, people can have different interpretations of what they see because they're sampling a small bit of that phenomenon or that object. And in this case, in our work, it is the neural activity that records from the brain. So I'm going to go over some vocabulary which I will use during my talk so that I avoid confusion. Ictal refers to the time of seizure, and length of the seizures differ between patients and even for each patient. Interictal time is the time at least 1 hour from any seizure. Epileptogenic zone or the network, epileptogenic network, are the areas primarily responsible for generating seizures. Seizure onset zone or network are where seizures start from. Epileptic activity is the pathological activity which might underlie seizure generation in epilepsy. And the localization which I referred to in this talk is to discriminate between pathological from health. It is the discrimination between pathological and healthy brain areas. So epilepsy is a brain arrhythmia, basically, where brain cells send the wrong signals. And the clinical hallmark of epilepsy is repeated seizures associated with and the associated comorbidities, risk of injury, and death. Epilepsy affects 150,000 patients in Australia and more than 50 million worldwide. And epilepsy can be caused by for different reasons and can manifest itself in different presentations and any age. And medications fail in 30% of cases to control seizures. And the patient's best chance would be surgery, in which clinicians try to localize where the seizures start to remove that safely without affecting cognitive, with minimal effect on the cognitive performance or the motor performance of the patient, as well as perceptual performance functions. So EEG is one of the critical tools that people use trying to localize this epilepsy, and we know what it is: recording of electrical activity in the brain. Also, MRI is used to find where we see pathology in the brain and the brain structure to see where it potentially could cause seizures in the brain. And sometimes nuclear medicine is used to find areas of the brain where we have difference in activity from the rest of the brain, whether it is hypometabolism or hypermetabolism. When these modalities fail, people try to use stereo EEG, which is the implantation of electrodes inside the brain to see where the seizures start from. And people are implanted with electrodes based on hypotheses where two electrodes should be put and implanted, and they stay in the hospital for 1-2 weeks to record activity from different brain areas. So hypotheses are developed about where to put the electrodes, because the number of electrodes cannot be unlimited. You have to put a limited number of electrodes, and based on what you see in MRI and EEG, surface EEG, you develop some hypotheses to test by putting those electrodes, implant those electrodes inside the brain. So signals are collected for 1-2 weeks and then are interpreted, and some qualitative features of those signals have been recognized based on decades of research and decades of clinical work, such as low voltage fast activity, DC shifts in the signals, rhythmic activity, and bursts in the signals, which might indicate where the seizures and which electrodes are showing those activities to localize the seizure. So, and then they proceed to resection or ablation or other methodologies to deactivate the brain area. However, still 30% of this procedure, people who undergo epilepsy surgery fail to become seizure-free. And one major problem is the difficulty to discriminate epileptic patterns from healthy signals. And that is because epileptic patterns are very complex, infrequent, and often different across patients. So, for example, you can see one patient implanted with electrodes here. You get to collect signals like these, and you have to discriminate whether the signals on these rows are pathologic signals or are these the pathologic signals. So it can give you totally different views about where in the brain the pathologic signals start from, and how they are propagating potentially across the brain. And it is only 40 seconds of data I'm showing you, but the clinicians and this epilepsy SEEG procedure collects about more than 7,000 minutes of data, where clinicians need to go through all those data to find pathologic patterns. One solution to this difficulty might be the quantification of signals, to get some measures out of those signals to compare those measures. So the quantification of signals is when you have a signal, you extract some features from the signal, and you get a value. For example, here, the variance of this signal is calculated through this feature extraction process, which is basically is like putting a lens onto a signal and looking at very specific aspect of the signal. Remember the elephant example. So I'm going to show you some signals with different levels of variance. So you can see variance is easily calculated. You can see the formula here, and you can calculate the variance of these two signals, and you can see that the top signal has a higher variance than the lower signal, which is indicated by these like higher peaks and valleys in this signal. You can see two signals of different complexity as measured here using Katz's fractal dimension, which is a measure of complexity of the signal to see what subsequences of signal, what subsequence of patterns, what subsequence of patterns are occurring in the signal. So the top signal here shows a signal with the Katz's fractal dimension value of 1.12, and the bottom signal shows a signal which is the same signal as the top one, except for a few points in time where I've flattened and where I've put a constant value in the signal, which you can see that the value of the complexity decreases, meaning that the bottom signal is less complex than the top signal, because the bottom signal you can predict one value from the other values, because it has less variable values and less variable patterns in the signal. So you can use those measures to potentially discriminate signals from one another. So this is a good review, which reviews the different ways you can quantify signals and potentially localize seizure in the brain. So I'm going to give you some examples of the interictal quantification efforts in the field. So basically two of the main signatures of seizures and epileptic patterns have been the interictal spiking as well as high frequency oscillations. So okay. So you can see here, for example, a spiking occurring with some high frequency oscillations recorded in rats, as well as in the hippocampal in the hippocampus of a human, which you can see shows a frequency of around 300 hertz. And also you can see a spiking recorded in entorhinal cortex, as well as hippocampus. And you can look at the zoomed version of the hippocampus activity, and you can see that there are some co-occurring high-frequency oscillations with the spiking, which you can zoom in and see. And these pathological patterns have been associated with the area of the brain which potentially generates seizures. And there is a debate between whether spiking activity or high-frequency oscillations are more indicative of the area or of the location of the seizure. And it has been shown that the co-occurrence of epileptic spiking as well as high-frequency oscillations give a better prediction of the location of the seizure, where the seizures will start from. But remember that these are recorded between seizures. Now I'm going to show you data from one patient we recorded. You can clearly see that the spiking is obvious across many different contacts in this seizure, in this patient. And you can see that, for example, some spiking might not be easily visible. For example, this one on an electrode, which might be more subtle than the other ones, which you might miss, but you can use some spike and HFO detectors to find those patterns and detect. Then you count the number of spiking activity across the different electrodes to find where potentially seizures start from. You can see the distribution of those spiking, co-occurring spikes and HFOs across different channels. And then what you can do is to check for the delay between those spiking activities across the cortex to potentially get an idea about where those spiking activities start and how they are propagating across the cortex, which we did in this study, and we found that they seem to potentially propagate from the N electrode, which was a mid-singular area, which also was where seizures started from. Other people have done other work as well, worked on the connectivity relative to this resection zone. So these are retrospective studies, and they have outcome for those patients as well. So you can see, for example, here that the connectivity between the region which has been resected, which has been potentially hypothesized to be the seizure onset zone, is higher within that zone. And it is seems to be higher than the other areas outside of the seizure onset zone, and it is higher in patients who got a better outcome of the resection than those who didn't. Which means that areas which generated seizures had more connectivity inside, and they were potentially less connected to other areas, and those patients who were resected, the seizure onset zone from whom it was resected, they got a better outcome than those who had less distinctive seizure onset zone from other parts of the brain. So you can also see that pattern here, for example, across different electrodes, the areas which were resected, when they correlated, the areas which were resected corresponded to the areas which give the highest level of within area correlation. They got better outcome compared to when the resected areas were different from those which had other areas which were more correlated. So people have, potentially clinicians have missed those areas. That's why they didn't get a good outcome, these patients. So, and other metrics, for example, they have shown that normative power can also be informative in localizing where pathologic patterns of, in the brain are generated. So they have developed these from electrodes which were in the non-pathologic brain areas. They developed across a group of patients. They developed these atlases of power across different brain areas and in different frequency bands, as you can see here. So these are normative atlases. So any new patient for whom we get powers in different frequency band, different from these like healthy patterns, can be considered as a kind of like those areas can be considered as non-normal and pathologic, and then these areas can be removed. They have shown that this method works, and in patients in whom the pathologic areas recognized by using this method have been resected, they got a better outcome than those who people targeted and resected other brain areas which were not non-pathologic based on this method. So they seem to have got good predictive power in terms of outcome as well. And I'm going to show you some ictal quantifications as well. So now we are going to the seizures to see what patterns seizures show. So a sample seizure, you can look at this pattern, but it might be a bit difficult to see actually where it starts by looking at the patterns electrographically and which electrodes show the first initial patterns of epileptic pattern, seizure onset pattern, basically. So this is the time where we roughly decided was corresponded to the seizure onset time. And you can see, if you look at closely at these electrodes, you can see that there are high frequency oscillations, high frequency, low voltage, high frequency patterns in these N electrodes, which develops and potentially propagates to other areas involving other areas of the brain. So this is one of the key features of epileptic. It has been suggested to be one of the key features of epileptic patterns during seizures. So low voltage, fast activity. And we can map them on the brain and find out where those electrodes are using some epileptogenicity maps and localize where the seizures start, which in this case was mid-cingulate cortex. So the idea of epileptogenicity goes back like 15 years back, and in this study, they developed an epileptogenicity index, which basically measured the level of energy in high frequency bands relative to low frequency bands as an energy ratio, in which higher increasing the higher frequency bands relative to lower frequency bands reflects epileptogenicity. And they combined it with the time that this increasing epileptogenicity starts in the brain across different electrodes to give each electrode an index of how epileptogenic that electrode is. So you can see that this is the metric. It combines the time when the increasing energy starts and how much energy increase we see. So this combination gives you a good measure. So, for example, this was tested on mesiotemporal patients with mesiotemporal seizure onset zone. You can see that the index was higher in mesiotemporal areas compared to lateral structures in the brain. And they recently developed another metric they called epileptogenic zone fingerprint, which suggests that the start of the seizure is reflected in three main features. One of them is the spiking of activity. This is the time of the seizure onset. Spiking activity before the onset, accompanied by fast activity upon the seizure onset and some slow wave shift which co-occur. So they have shown that sharp transient spikes, multi-band fast activity, as well as suppression of lower frequency. You can see that in the time frequency plot as well, spiking, increasing power in high frequency, and decreasing power in lower frequency bands. It has been shown that it was a successful measure in predicting finding epileptogenic contacts and epileptic seizure onset zone, and got good results. Okay, so also people use node centrality in gamma cross power spectra, in which they calculated the connectivity between different electrodes upon the seizure onset, and then they extracted the eigenvalue out of these adjacency or connectivity matrices over time, and found the electrodes which gave the highest centrality, meaning that those electrodes potentially were the most central, most connected to other structures upon the seizure onset. And they found that this metric could also predict the outcome of those patients who were resected, whose resection overlapped with the areas which they predicted to be the most central part and the most critical node in the network. So these are the methods they have used, but it has been also shown that epileptogenic patterns and especially seizure onset patterns are not just one, but rather a variety of different patterns can appear across different patients. So for example, you can see that low voltage fast activity, which is the most critical one based on the literature, and also we can have predictable spiking as well as burst of polyspike followed by low voltage fast activity. And in general, low voltage fast activity has been one of the most common signatures of epileptogenic zone, and sometimes it is accompanied by other patterns which have been observed in other patients. So, but importantly, there are a variety of different patterns, and LVFA only accounts for 45% of cases, and sometimes it shares other characteristics with other metrics, but still it doesn't seem to be a comprehensive signature of epileptogenic zone. So in the first study, we tried to answer 3 questions which we thought were worthy to be investigated. And we thought that we might need to look at multiple signal patterns based on the previous slide that might be important for localizing the seizure onset zone. We also thought that we don't know how much intrictal the patterns, epileptogenic patterns between seizures generalize to the time of seizure. It is important because not all of the patients who are in the hospital are and monitored for epilepsy produce seizures. So it is important for you to be able to use intrictal patterns, whatever that is, to be able to get some localization power. And we wanted to know how much correspondence there is between these interictal patterns and ictal patterns, and what features of those activities are shared between the 2 time points. And we were also wondering which epileptogenic patterns systematically generalize across patients, rather than finding a specific pattern which is successful for one patient but not the other. We wondered how much of which patterns generalize across patients. So let's do some discrimination of patterns together to be able to show you the value of having putting different lenses on the signals. So if I ask you to tell me if these 2 signals are identical or different, what do you think?
I would say identical, and I can't wait to be wrong.
Okay. So if you put a few lenses on this signal to quantify it, you potentially, several of these measures also tell you that these are identical. There is no difference between them. The y-axis shows the difference between them. And these are the lenses that I put on the signal, the ways I have quantified the signal, which are mostly based on the complexity of these 2 signals. However, if you increase the number of lenses you put on the signal, you can see that these two signals are different from specific viewpoints and from specific aspects. So the median value of these two signals are different, and other metrics of these signals, such as the skewness of the signal and other metrics and some frequency specifics. It is just that the lower signal is shifted upward just a little bit. And if you look more closely, you can potentially see that from comparing these two peaks. So adding more lenses onto the signal, quantifying them through more metrics or measures, you can potentially see the difference between signals. And the metrics I've calculated, extracted from these signals I've not so strictly categorized them into moment features, complexity features, and frequency features. Okay, let's look at another signal. Do you think these are identical or different? I guess it is clear that they are different, right? And many of these features show that these two signals are different. But it is also interesting to note that some of these features do not discriminate these two signals very like robustly or clearly. So some features might not pick up the difference, some features may. If you look at these two signals, what do you think about these two?
I think it's identical.
Yeah, they are identical. Okay, what about these two?
Same.
Let's see. Many of features between these two signals are different, it seems, except for a few. So what is the difference between these two? There is only one sample different between these two signals. So you can see that the median of the signal hasn't changed. The sample hasn't changed the LZ, LZ complexity of the signal, but many other factors have changed. The average frequency here is determined by how many zero crossing we have in the signal. So you can see that you may miss it. And this is the challenge that people have in epilepsy monitoring. So a multi-lens view and investigation might help us in recognizing the differences between signals. So if it is the signal dynamics that we are looking at, if it is the whole dynamics of the signal that we are looking at, which is like the whole elephant, when we look at a specific aspect of the signal, such as the mean of the signal, we are looking at a small portion of the characteristics of that signal, which might not cover the whole thing and might overlap with the next feature that we extract from signal. But importantly, there are areas of those features which do not overlap and which adds to what you can see in the signals. But so with adding more feature or extracting more features, you can potentially cover more and more of the characteristics and the pattern in that signal. For example, if you look at the sample entropy, it might not overlap with the other two features, but still helps you to cover more of the signal dynamics and capture that dynamics. Some other features may overlap, some other features may not. So by adding more and more features, you potentially are able to capture as much information, as much dynamics of the signal as possible. And in the case when you're trying to discriminate two signals from one another, there are signals sometimes overlap with one another in certain aspects, but are non-overlapping in some other aspects. So it might be useful. So when you look at a specific aspect of these two signals, you might be looking at some overlapping areas of the signals. But by using other features of the signals, which do not overlap with one another, and look at the specific aspects of the signals which do not overlap, you might be able to discriminate those signals better. Okay, so we potentially can increase the number of features and improve our discrimination. And what about when we have many signals, like many patients? Potentially, machines can help us discriminate those patterns. Let's look at one example. Here, I'm trying to train a machine to discriminate seizure versus normal patterns of signal from one another. I need to train a machine and then test the machine with new data. So these are epileptic signals and healthy signals. So imagine that I extract frequency features from signals as well as the variance, which is another feature of the signal. So this signal corresponds to one point in this two-dimensional space. Another epileptic signal, another epileptic signal, another, one more, and some healthy signals. 1, 2, 3, 4, 5, 6. So you can see that these two patterns clearly can clearly be separated. So the machine's role is to find that hyperplane which separates these two patterns. And anything from there then on, anything that falls on the right side will be considered as an epileptic signal, and anything on the left will be considered a healthy signal. Now let's test the machine. You give it an epileptic signal, it falls on the correct side, a healthy signal, epileptic, healthy, epileptic, healthy, epileptic, healthy. Sorry, I think it is miscolored. But it was supposed to be an epileptic sorry, it is an epileptic signal. I was just saying that incorrectly. But anyway, one of the patterns fell on the incorrect side of the hyperplane, which means that it has been misclassified as an epileptic. So a machine is not perfect, but it can potentially discriminate signals from one another in the high dimensional space. And we tested this machine learning approach on an open source data set. The data set here includes 55 patients with focal seizure onset zone. Sorry, it has patients in a SEEG with SE implanted with SEEG, which is the implantation of deep electrodes, as well as ECOG, which are the grids which we put on the cortex. Seizure onset zone contacts are labeled in this data set by clinicians. Resected contacts are also labeled. Resection outcomes are available. We have only two 2-minute interictal epochs of data, as well as an average of 3.7 ictal data, 3.7 seizures from each patient. So let's see what is the localization performance. So the localization here again means discriminating epileptic and non-epileptic patterns from one another, or epileptic and non-epileptic contacts from one another. So we trained the classifier 90% of data and tested them on and they left out 10%, cross-validated them. This is the result we got, which is relatively good. So we did it within each patient. So 90% of contacts as training, 10% as test. For the interictal data and for the ictal data, you can see that we can clearly discriminate the two sorts of contacts, pathologic and healthy and or normal signals in both time spans. And interestingly, it is quite high in the interictal using interictal data as well, which is not very common in the field to see that high of a percentage. So the effect of we then look at the effect of surgery outcome, whether the patient was seizure-free or not. And the region where the seizures started from, the pathology, whether it was lesional or non-lesional, and the recording modality, which was SEEG or ECOG, on the performance. We didn't see any effect of those metrics on the classification performance, which could, to some extent, be explained by the very high percentage of percentages. But anyway, in terms of contribution of different signal features to localization, we wondered which signal features contributed most to the discrimination of patterns from one another to provide insight for the clinicians. So what we saw was that there is variable contribution. So the y-axis shows the contribution of each feature to the classification, and the x-axis obviously shows the different features that we use in the classification. What we see is that there are different levels of contribution from different features, obviously, but it is not like a significant difference between some features versus other features. And there seems to be some correlation between the interictal data as well as the ictal data, which you can see here. And these two were correlated, meaning that roughly similar sets of features could contributed to the discrimination of epileptogenic and healthy signals from one another. But importantly, there was no clear domination of specific features for discriminating, which means that potentially individuals differ in terms of what patterns of pathologic patterns indicate their epileptogenic zone. So we also wondered whether epileptic patterns generalize between interictal and ictal. As I mentioned, it is important to be able to, without looking at seizures, be able to localize the areas which potentially produce those seizures. This is the performance that we found. For this analysis, we trained our classifiers on the interictal data and tested them on the ictal data to see how generalizable the patterns were. And we also did the reverse because we had more ictal data. We thought that maybe training on ictal data would potentially improve the classification. What we saw was that the classification was above chance, but it was not as good as when you do it within interictal and separately within ictal. And it suggests that the patterns of activity between interictal and ictal do differ from one another, and they might not be very generalizable to one another. And it is important to note that above chance generalization was not guaranteed. So it could be the case that the areas which generate seizures had a lower power in between seizures, and they would start producing more power than the rest of the areas during seizures. So in that case, we would have seen below chance decoding or generalizability. But here we saw above chance, which means that there is some relationship between interictal and ictal data, but the relationship is more complex than can be captured easily with a simple generalization platform. And we saw that similar sets of features generalize in both directions, which means that when you train the classifier on interictal data and predict ictal data, similar sets of features contribute to the generalization, as well as when you go and the other way around. Then we looked at cross-patient generalization, which is important for real-world application of the method, because ultimately you need to train a machine to learn all the epileptic patterns and then test it with the new patient's data to see how generalizable those patterns are and which patterns are generalizable across patients. So we trained the machines on all our patients except for one, and then tested them on the left out patient and cross-validated the results, the procedure. What we saw was that in interictal activity, in both cases, interictal and ictal, they were above chance performance. So you could predict it with some accuracy, but not very good. But in the ictal activity, we had better predictions, about 75% on average for patients to be able to localize the seizure onset zone by looking at the previous patients. So there were some commonalities between the patterns of activity ictally compared to interictally. Interictal activity seems to be more different across patients compared to ictal activity. Okay. So then we looked again at the contributions of the features. We saw a different pattern here. We saw that the patterns, the generalizable patterns, which the features which generalized in the intracranial data differed from those which generalized in the ictal data. For example, if you look at the complexity features we have here, you can see clear difference. So these complexity feature, approximate entropy, Heort complexity, and Heort mobility, these features were more informative for ictal data compared to interictal data in terms of their comparison with other complexity features. But more importantly here, if you look at the power of the signal in different frequency bands, you can see that as opposed to the literature, which suggests that high frequency power is the more dominant and more reproducible pattern across patients, mostly based on qualitative assessment, doesn't seem to be as generalizable as the lower frequency patterns, which potentially refers to slow shifts in the signals at the initiation of the seizures. And you can see that this is different for the interictal activity. Higher frequencies were more generalizable in the interictal compared to ictal activity. So we saw a kind of negative correlation, which was non-significant, but yeah, it was not related. So these two patterns, different sets of features, generalize across interictal and ictal across patients. And we saw that patients varied in their generalizability. So which means that if a patient's epileptic patterns are different from the rest of the patients, it probably reflects in both interictal and ictal data of that patient. It can be for different reasons, potentially their implantation. Please remember that the implantation for every individual patient is different. So one patient could have been implanted in one area of the brain, whereas the other patient could potentially have been implanted in other area of the brain. So there are all these complexities and differences that can explain that. But when a patient's signal is different, it is reflected in both interictal and ictal data. And we saw some advantage for the temporal versus mesiotemporal patterns in the generalization, which means that mesiotemporal patterns were potentially more similar to one another because they came from a more constrained area of the brain compared to the whole temporal lobe, the patterns that are generated across the whole temporal lobe. And also we saw that a clear advantage for SEEG implantation and generalization when using SEEG signals compared to ECOG, which is potentially explainable by the fact that SEEG usually records diverse sets of brain areas, potentially increasing the potential overlap between patients, compared to ECOG, where you have a grade which might or might not necessarily overlap with the other patient's data. And we also had a higher number of channels for SEEG compared to ECOG on average, which could potentially explain that as well. So the summary of study 1 would be a multi-feature or data-driven discrimination of signals has potential for detecting diverse epileptic patterns. And interictal and ictal features which generalize across patients differ, high frequency power and energy ratio, which were suggested by the previous literature, were not the most generalizable features in the ictal data. And SEEG detected more generalizable patterns across patients than ECOG. And now I'm going to start it will be a shorter study. So the second study is to look at neural activity, trying to see if directed connectivity across different nodes of the network can inform us of the location of the seizure, where the seizure starts. So we asked if there was a dominant direction of activity flow relative to the seizure onset zone between seizures in the interictal period, and if so, does the directionality change when seizures occur? And can these directionalities localize the seizure onset zone? Okay, some studies suggest that potentially in the interictal period, so this is the seizure onset zone, and these are the non-involved areas, they potentially send signals towards the seizure onset zone in between seizures to suppress it and avoid seizures, to suppress seizures and not let the seizures to start. Which means that inner strength, which refers to the number of links towards the seizure onset zone nodes in the network, are higher than the non-seizure onset zone contacts. But in the ictal phase, it has been suggested that the seizure onset zone, which is this area, more dominantly sends out the signal to different brain regions to initiate, to like spread the seizure to other parts of the brain, which means that the outer strength, which is the number of and the strength of the outgoing connectivities, are higher for the seizure onset zone contacts than non-seizure onset zone contacts. But what does the literature say in that? It is not, in fact, unanimous. So in the interictal period, seizure onset zone has been suggested to be in sender of signal. For example, this study localized the seizure onset zone using a source localization and found that the seizure onset zone corresponded to the sources of the signals using adaptive directed transfer function as a measure of directed connectivity. Another study showed that, again, this epileptogenic zone or the seizure onset zone sends out a signal during the interictal period using directed nonlinear regression as the measure. Also, seizure onset zone has been suggested to be a receiver of signals. For example, in this study, which used partial directed coherence as the measure of directed connectivity. Also, another study found the same, that seizure onset zone seems to receive the signals, and that's why the seizures do not start to propagate. And directed transfer function was what the method they used to measure these connectivities across the cortex. In the ictal period, again, we have discrepancy in the literature. Seizure onset zone is the receiver, which might be strange, but some studies showed that partial directed coherence shows that in degrees higher for seizure onset zone compared to in degrees higher for the seizure onset zone compared to the other areas. Also, another study found that a seizure onset zone seems to receive signals from other areas at the start of seizure using Granger causality analysis. Again, another study found that seizure onset zone seems to be the sender of signals at the start of seizure, which is here, and the other areas seem to be receivers of the signals. Again, another study which used directed transfer function found that it is the case. Seizure onset zone seems to send out the signals during seizure. So we can see that the results are discrepant. And one potential reason for the discrepancy is that people are looking at different, using different measures of connectivity to check for the propagation of signals across the brain. So we thought that we could test these two hypotheses. The seizure onset zone receives the signal in between seizures. That's why it doesn't seize. And seizure onset zone sends out the signal during seizures because it is in a refractory period and starts to send out the signals. So in order to do that, we extracted the signals. We look at different windows of the signal, and we extracted different measures of directed connectivity. We used 13 different measures of connectivity from PySpy toolbox, which is developed in your lab. And so these measures measure the levels of signal propagation from an area to another area. And we use 6 different node metrics, which refer to in and out strength of signals which includes in and out strength of signals from brain connectivity toolbox to see the general patterns of general roles of different nodes of the network and see how they behave in between seizures and during seizures. So we saw a higher flow towards the seizure onset zone intrictally and away from it during the ictal period. So these are the results from the different connectivity measures or different lenses that we put on the signals to test the interictal, whether there was a higher inner strength. So you can see that many of these metrics show higher levels of inner strength signals going towards the seizure onset zone compared to non-seizure onset zone. And not all of them were significant, and there were a few metrics which were not supporting that, but almost half of them supported that. And also in the ictal period, we saw that it was less unanimous than the interictal period, but some of the measures, such as these two, showed that information was going out of the seizure onset zone and propagating to other parts of the brain, basically. So it seems that if there is any support, the evidence seems to suggest that signals go towards the seizure onset zone between seizures and out of the seizure onset zone in between seizures and out of the seizure onset zone during seizures. But we also asked, does the direction switch from interictal to ictal period at individual level? What we observed was that 5 of the measures showed that signals, so for the participants, for the patients in whom there was a higher degree of ingoing signal interictally, there was more outgoing signal ictally. That's why we got these positive correlations for several of our measures. Other correlations were all non-significant, meaning that they were not, they didn't show the opposite effect, but they were non-significant. For those which were significant, they all showed that patients in whom seizure onset zone suppressed between seizures was propagating more during the seizures. Okay, we also asked whether the seizure onset zone was most central or peripheral in the network as a whole. To address that, we extracted different sets of metrics from the nodes, such as the first passage time, clustering coefficient, eccentricity, and betweenness. So a higher first passage time refers to more peripheral nodes. And we focus also on the eccentricity, which gave us more unanimous results. The other two didn't give us a clear result in just one metric showed higher and the other one lower, so they were not unanimous. But the 2 other metrics, which referred to how central the node was in the network, showed a relatively unanimous results, and they suggested that signals seizure onset zone seems to provide a higher first passage time compared to non-seizure onset areas by several metrics, and there was a higher eccentricity for seizure onset zone compared to other areas both in the interictal period. In the ictal period, there was less, the effect was smaller, but again we saw some evidence of information higher ictal passage, first passage time as well as eccentricity in the ictal period. Both of these suggest that the seizure onset zones seems to separate itself from the rest of the network. And it is important to note that this separation of the seizure onset zone compared to other parts of the brain is not a spatial separation, but rather a kind of connectivity-based separation, so that it doesn't seem to, it's the activity that it produces and how it interacts with but with other parts of the network seems to be different from how other parts of the network interact with one another. So we try to localize the seizure onset zone using these metrics, directed connectivity metrics, as well as the node metrics that I presented to you, to see how powerful and how consistent these directed connectivity measures are for localizing the seizure onset zone. What we saw was that it is not super powerful, which means that we got a median accuracy of about 65.65, which refers to the discrimination of seizure onset zone versus non-seizure onset zone contacts, both in the intra-ictal as well as ictal period. But when you're looking at the contribution of the different connectivity metrics in this classification of signals, we saw that directed information, which is one of the information theory-based connectivity metrics, showed a higher contribution to localizing the seizure onset zone compared to other metrics of the directed connectivity network and directed connectivity metrics. And we also saw that the eccentricity of the seizure, the eccentricity measure provided more information compared to other measures of node behavior in the network. So the node being less central seems to provide information about where the seizure onset zone is. It was also relatively similar in the ictal period as well, with directed information playing a more, let's say, pronounced role, as well as the eccentricity playing a more dominant role in the discrimination of the seizure onset zone from the rest of the network. So we checked the stability of the network measures over time. In the interictal period, there was not much variation in the connectivity measures over time, because patients were in resting state, and not much was happening and changing. But here, during the seizure, at the beginning of the seizure, there were more discriminability for the seizure onset zone. It was easier to discriminate the seizure onset zone from the rest of the network at the beginning of the seizure, which is compared to the later time points in the seizure, which is somehow explained and supported by previous studies, which showed the discrimination of seizure onset zone from the rest of the network using non-directed connectivity measures. So the summary of the second study is that not all directed connectivity measures supported source and sink seizure onset zone hypotheses, but some did. And there was some consistency in directed connectivity patterns, which allowed for localization of seizure onset zone. It still needs improvement, though. And seizure onset zone separates from the rest of the network, especially in between seizures. And the excitatory and inhibitory nature of the directed connectivity measures that we calculated is not clear yet. We need more information to be able to see if the nature of the signals which are going towards the seizure onset zone are inhibitory to inhibit the seizure onset zone or not. And with that, I would like to conclude my talk. So SEEG procedure, implantation of electrodes inside the brain to localize the epileptogenic zone, is a growing approach in epilepsy surgery globally as well as in Australia, and it can be improved through quantitative analyses. And open source signal quantification toolboxes such as CAT22 and PySpy, which have been developed in your lab, allow for more exhaustive evaluation of neural time series. And data-driven methods provide additional insights into the brain by adding more lenses onto the overlooked aspect of meaningful neural activity. And with that, I would like to thank you and all my collaborators, especially Eileen McGonigal, who leads the epilepsy unit, for their efforts and discussions throughout these projects. And I'm happy to take any questions you might have. Thank you.
Thank you so much, Hamid. What a fantastic talk. And throughout, I loved your analogy of the multiple lenses and your different diagrams of signal dynamics.
Thank you.
I think we talk a lot in the group, which I'll open it up to in a moment. We talk a lot about finding this happy medium between data-driven analyses and a more constrained hypothesis-driven approach and kind of threading that needle. And I think all the work that you presented really struck an elegant balance there. So thank you very much.
Thank you.
I have questions, but I'll open it up to the floor first because I'm sure people are very interested in the work.
Sure.
Yeah, I have some questions. And first, I very appreciate your work because you introduced many index I haven't thought before, such as epileptic zone fingerprint and the epileptic index. So, so can I ask a fundamental question first? So, in the concept page, you introduce a epileptic zone, which is generating the seizure, and the seizure onset zone, which is where the seizure starts. So when you are localizing the SOZ, are you talking about EZ and SOZ both, or you are trying to distinguish them?
So that's a good question. So honestly, it's a difficult. So it is. They are defined. These two, SOZ and epileptogenic zone. These are defined in the literature, but they are often used interchangeably. So the reason I mentioned both of them at the beginning was that when I show the literature and I talk about the results of previous studies, when you see EZ, when you see SOZ, you do not get confused. But they are most of the time used interchangeably, but with I think Aileen would be a better person to talk about it. She knows the literature, but my understanding is that they are most of the time used interchangeably.
Yeah, I think so.
When I talked about seizure onset zone, I was talking about seizure onset zone, the area marked in the data set as where the seizures started from. But other people have used them interchangeably. Yeah.
Yeah, good. Thanks. And I have another question. It's about your study one. So you're comparing this index within patients and cross-site and cross-patient. And in the cross-patient study, this index in contribution is very different in interictal and ictal area. So is there exists index they are both has high contribution in interictal and ictal periods?
Sorry, are you asking which feature contributed to both interictal and ictal simultaneously?
Yeah, when you are comparing cross patients.
I was more looking at their discrimination, but yeah, we can go and look at them to see which feature contributed to both simultaneously. I think variance of the signal and the complexity. Several measures of complexity, I know that they contributed to both interictal and ictal, as well as complexity measures. I think they contributed to both relatively similarly, but as you saw, their contributions were not perfectly similar.
Yeah, thank you. And I have a last question. Can I ask? And it's about your study too. So you are comparing different measures to trying to define which which node is more behaving like a sink in interactal, it's the node is more behave like a source node in ITO period. So these measures are based on the connectivity, but I'm interested about does the connectivity itself can reveal the sink or source function? Just wondering, have you prepared it?
So the way we try to infer whether a node is a sink or source was through the network metrics of inner strength as well as outer strength. So inner strength for the interictal period.
Yeah.
So higher inner strength, which would refer to higher, larger connectivities connected to incoming connectivities to a node, and higher outer strength during seizure, higher outgoing connectivities. But within a connectivity measure, we didn't look at the every individual. I think it would be difficult. That's why I think we used these like connectomics metrics to sum up what we could see from what we saw from the seizure onset and non-seizure onset zone. So they would summarize what's happening there instead of you looking at every individual node connections to other nodes. I think that was the main reason for us to use these connectomics metrics.
Yeah, thanks.
Thanks, Ruby. Joe, I see you have your hand raised.
Thanks. Thanks, Annie. Picking up on, I guess, some of what Ruby was asking at the end there, and that switch between, you know, the higher connectivity in the interictal versus higher connectivity out in the ictal. I'm wondering if you dug into what makes that flip. Is it that the incoming connections are becoming lower during the ictal, or the outgoing connections are becoming higher and that's what's overtaking the incoming ones, or is it a bit each way?
You mean are you asking about the strength of those connections or the number of those connections? We haven't looked at them, but I guess that inner strength would take into account the weights of those connections, right? But we didn't look into the number of connections, whereas the weights of those connections, if that's what you are asking.
Yeah, in part. I mean, if you come back on your slides, maybe it's on the previous slide to this or two previous slides or something, we might even be able to see it on the graph. Oh, no, sorry, when you first got the results, it must be after this. We just went past it. Yeah, here. So your conclusions are based on, for each measure there, the comparison between the red distribution and the blue distribution. And so I guess what we can do is we can look at, you know, how those red distributions compare in that, where are we? In the interictal.
Sorry, this is the interictal, this is the ictal.
Yeah. Sorry, I think have I misinterpreted? No, red is SOZ, blue is non-SOZ.
That is correct.
Okay, so we've got just in strength for interictal up the top, and we've got just out strength for ictal down the bottom.
Yes,
okay,
that is
so
correct.
I guess we can't see it from the graph here then.
That is correct.
I'm just curious. You know, is the difference changing because the in strength is going down, or it's because the out strength is going up, or it's a combination of the two, and whether that is more connections or whether that's stronger connections? I don't know. I was just curious if you dug into that.
Yeah, that's that. These are good points. We didn't look into that, but yeah, I think that's a good point.
Okay,
yeah.
That's okay. Just curious.
Yeah, that's a good point.
Can I throw another question at you while I've got you? I'm wondering about whether the question had come up for you guys about the validity of any of these statistics when you have highly non-stationary data. So clearly, when there's the ictal periods taking place and you've got a lot of instability in the network, you're going to have much more non-stationary data. And a lot of these measures, in principle, you're not supposed to use them on non-stationary data. As an information theorist, this is something I hear from people all the time when they get cranky about how we try and do things sometimes. Now, I don't think there's a problem here. I'm just wondering if you guys have received any pushback similar to that.
And we haven't got the paper reviewed yet, but we might get that. But I think so. We decided to not use very long time windows of data to make them more like a stationary signal. So these are 2 seconds of data and like 500 time points, 250 hertz. So yeah, I think I totally agree with you that during seizures, a lot of things happen, a lot of things change over time. But I think the way we were trying to address that was to shorten our windows. But we have, and we looked at different time windows to see if it changed, and we didn't. What we saw was that at this onset of the seizure, there was like more discrimination between seizure onset zone and non-seizure onset zone, and later it was like less pronounced. But yeah, please, yeah, it's good to talk to you about these details. Yeah.
Cool. Okay. Thank you. Very interesting study, by the way. Lots of very impressive work.
Thank you.
Fantastic. Are there any other questions from the room? I'm cognizant that I'd scheduled this until 1:30, so people might need to filter out, including you, Hamid. But if there are any last questions.
No problem. I'd be happy to.
I have a question. It kind of relates to what Joe was saying. So I'm curious about what you think is happening biophysically when a node in the networks are more of a source than a sink. Is there some type of local EI balance issue or change synaptic efficacy, or is it something more at the network level?
So what I have read is that intricately when the nodes, like when the seizure onset zone, the reason for seizure onset zone receiving the signals is that it is being inhibited by other areas. But the nature of that inhibition hasn't been shown, I guess, in the work that I have read. But no, I don't have a clear answer for that. But yeah, it's a good point maybe to investigate, see what happens there.
Can I riff on that theme a little bit with you? On a related note, earlier in the presentation, you mentioned that there was some evidence that the SOZs had a lot more internal connectivity, recurrent connectivity, I guess you might call it, than other zones. It would seem difficult to infer that from data. Here, for example, we're looking at pairwise measures, and it doesn't look like there's any obvious way to try and relate that to, those earlier findings about stronger internal connectivity. I mean, there might be, but it's just not obvious. Is that something you guys thought about as well?
Sorry, your point is, can you elaborate on that?
Yeah. So I was picking up on something you said in the first half of the talk from the background. I'm trying to find it in my notes here. There was
where they showed that seizure onset zone had a higher internal connectivity than areas outside of the seizure onset zone. That is correct, yes.
Yeah, exactly. So I was wondering whether that's something that we could measure from the data as well. I mean, here we've looked at pairwise measures, obviously from PySpy, but maybe that's something that comes up with other kinds of time series measures that might relate to it. Or not, or maybe it's something that, if you are seeing less connectivity incoming from other regions, maybe that signals higher connectivity internally. I don't know. I'm just wondering if you guys had thought about how these results related to that.
Yeah, that's a good point. So I think it is quite well established. It was not the only study that I presented which showed that there was a higher connectivity within the seizure onset zone. There are a lot of them having shown that. So we guessed it was, we assumed that that was the case. Do you see any relationship between the inner strength? So are you suggesting that higher inner strength during the seizure, during the intracranial period, can potentially mean a higher internal connectivity within the seizure onset zone?
I'm not really sure what I'm suggesting. I'm just trying to work out if there is a relationship between that higher internal connectivity and some of the pairwise measures that we see here. Like maybe there's often you see an inverse. If internal connectivity drops, incoming external connectivity can go up. Often in other systems. So maybe that's something similar here. Just
yeah.
Yeah,
So
no.
what I also did was to so these ones are between every seizure onset zone and all the other areas, even within the seizure onset zone. What I did was to get an average of all the seizure onset zone to other external areas and saw relatively similar patterns to what we see here. But I didn't look specifically at internal connectivities within seizure onset zone and outside of it to see the relationship between them. But yeah, most of the work that I know, which looked at the seizure onset zone connectivity, showed higher connectivity. They use undirected connectivity. Directed.
Yeah, that does seem quite problematic, doesn't it? To be coming at it with a lot of undirected measures. So yeah, it's definitely good to see the directed work here.
Yeah.
Great, thanks. I might ask one final question if that's all right, since
Sure.
Directed connectivity and the relationship between local dynamics and pairwise coupling is something I'm really, really interested in my research. So I might pick your brain for a final project in that. I'm wondering with some of the SPIs in PySpy, a larger value will indicate greater information flow or greater coherence from one time series or one region to another, like directed information or directed coherence. Whereas for others, the value of the SPI actually reflects less information flow or less coherence, something like the mean squared error of a linear regression fit for the LM fit and Recce regression error causal inference SPIs. And I'm curious how you've reconciled this kind of difference in the positive or negative relationship between these when computing nodal properties here.
Yeah, that is correct. I think one of the other reasons we decided to use these connectomic metrics was to avoid that kind of comparison. We wanted to see some sort of rather than the value of the connectivity itself, we wanted to see the behavior of the node relative to other nodes in the network in terms of comparison with seizure onset zone in the strength and non-seizure onset zone in a strength compared to one another, rather than looking at the absolute value of the connectivity, how it changes. And I think, I think that was the reason, to avoid those hard to interpret effects.
Gotcha. Thank you. Well, if there are no other burning questions in the room, please join me in again thanking Hamid for his time today and for a really fantastic talk.
Thank you. Thank you very much for the invitation. It was great to talk to you guys.
Great. I'll stop the recording here.